Um, Good evening, dear students. I welcome you all to another class of the chapters that are going to follow after plant morphology. All right. So, hello, Government Meso School is all. How are you? Hello, everyone. And hello, Shillong Center. How are you, students? I'm very happy. I got let's say, quite a few messages from you all. And I was very happy to see what you people think about yourselves and your hopes and dreams and aspirations, all of these. Look, I will try my best to answer your messages, and a few messages are still to be replied to. I'll try my best. My schedule was very busy yesterday and day before yesterday also. And just today, right now, I checked some messages right now. Okay. So, as soon as I get time, I will respond to all of your queries, everything that you people are asking. And I will ask all of you to continue, okay? So, because it's very important for me to know your names, to know what you people think about yourselves, about biology as a subject, my subject, okay? So, because biology is the main subject, you all people know that. Biology is your main subject, and just like maths is for math students and biology is for bio students, okay? So, please respond to me, okay? And we'll talk about a lot about your life, my life. You can ask me things about my life also. I don't mind that. And in this way, we'll continue, and we'll also cover this syllabus that is remaining, okay? So, please remember that the class 11 syllabus is quite lengthy as compared to class 12th. So, from now on, I have made a small change. I'll be running some presentations, okay? And after the class, you can take these presentations from me, okay? I had prepared some presentations, and I am supposed to make some more changes to that. So, we'll today continue with the plant anatomy, and I see only 3 centers online right now. Government Meso School, what's your attendance today? How many students are present today? Government Meso School, please respond. I believe you can see me, right? I'm not invisible, right? So please tell me what's your attendance. And Shillong Center, how many students are present today? Yeah, you can hear me, I think. All right. Hello, St. Paul's. Hi to you all. 7 students, great. All right. And how many students are present in St. Paul today? Please share your attendance. I would love it if instead of just writing your attendance, it would be very good for me if you people wrote your names, okay? If you people wrote your names in your attendance, it would be very fruitful for me, okay? Because then I can recognize by your names, because you people send me messages, and I also know you somewhat. I am beginning to know you. So it will be 2 students are present here, okay? All right. Hello, Vankar, how are you? Hello, Gazania, how are you? Yeah, that's great. Please share your names, okay? I can. Hello, Silver, how are you? I saw your messages. And hello, Diana, how are you? I now recognize you, okay? So this is much better. If you people write your names, then I can recognize the people who have sent me messages, who are talking to me. And hello, Dayahun, how are you? I recognize you. And I recognize Diana, I recognize Vankar, and I do not recognize Betwan Sao. Okay? All right. So it will be very helpful for me if you write your names, because then I can address you by your names, not just in front of the camera. All right. So let's begin our today's session. Okay. So I was talking about plant anatomy in the last class also. I had finished with morphology. I will give you a presentation about plant morphology, the root, stem, leaf and everything. I'll just prepare it fully and share it with you on the group, okay? And continuing with plant anatomy, I'm going to share some slides, okay? And just to give you an idea what we are going to do today, okay? So I was talking to you about plant anatomy, and the first thing that I told you about was tissues, okay? What is anatomy? We study the arrangement of tissues, okay? What is anatomy? We are studying the arrangement of cells or tissues, okay? Arrangement of tissues in the plant body. This is not an exact definition. You do not have to exactly write this, okay? But it's much more accurate than what is written in your NCERT. Arrangement of tissues in the plant body, okay? Plant anatomy, the definition must be given like it's a simple internal structure. But what do you mean by the internal structure? By internal structure, we mean the arrangement of tissues in the plant body. So next the question comes is, what is tissues? You already know that tissues means the group of cells which have similar origin and similar function, all right? So I have already told you about it. Then after tissues, we talked about the types of tissues, okay? Now to classify anything, there are many parameters, okay? There are many parameters to classify anything. So about tissues, we can say these are simple tissues, these are complex tissues. Simple tissue means the tissues which are composed of one type of cells only. Complex tissues means tissues which are composed of two or more types of cells, okay? So this is the one parameter. Another parameter can be ground tissue system or vascular tissue system. Another parameter is meristematic tissue or simple tissue. Lots of things, okay? So one parameter is simple tissue and complex tissue, okay? Under this parameter, we have in simple tissue, we have parenchyma, we have sclerenchyma, we have collenchyma, all right? In the complex tissue, we have basically xylem and phloem, okay? So parenchyma, sclerenchyma, collenchyma, this is one parameter. Xylem, phloem, one parameter. The another parameter can be meristematic and permanent. Another parameter is meristematic and permanent. Hello, Nahalagun students. You are online too? Yeah. Very nice to see you. Centers, please report your attendance, and the students who know me, who are talking to me, it will be better if you write your names, okay? I would love it if you write your names, okay? So please write your names. Nahalagun students, those who know me, and I know you a bit as well, okay? All right. So another parameter of classifying tissues is, do not worry about writing all these things. I will give you the notes, okay? So just concentrate. Hello, Nabam. How are you? I do not remember you by the name, but you just drop me a message separately, all right? You were recently added in the group, I saw. Yeah, I know that you were recently added in the group. All right. So tissues, another parameter is meristematic and permanent. Meristematic and permanent. This is another parameter, okay? By parameter, do you know what parameter means, okay? Do you know what parameter means? Parameter means we are choosing a criteria for classifying something, okay? And by choosing a criteria or by choosing a parameter, I mean like if you have a large number of friends, okay? So you can classify them as this one is more useful friend, this one is a friend I can count upon, and this one is a friend that I cannot count upon, okay? Another parameter can be, suppose there is a friend who you cannot count upon, but he likes to give parties and all. So you can always ask him for money or something like that, or you can ask him for a ride or something on his bike or anything. So there are many types of parameters by which we like to classify people, things, everything, okay? So the scientists also have parameters to classify the tissues, that is, okay? So tissue, simple, complex, simple tissue, parenchyma, sclerenchyma, collenchyma, complex, xylem, phloem, you already know it. Tissues, another parameter is whether the cells are dividing or they are not dividing. A non-dividing cell is a permanent tissue, a dividing cell is a meristematic tissue, okay? And in meristematic tissue, we have further some divisions, okay? Shoot meristem, shoot apical meristem, we call him SAM, okay? So shoot apical meristem is SAM, and root apical meristem, we can call him RAM. Like your mobile phones must be having some form of RAM, right? 1 GB RAM or 2 GB RAM, okay? Or 4 GB RAM. In the most advanced phones, it is a 4 GB RAM, or even some cases in iPhone we have 6 GB RAM as well. And I have heard about a phone which has 8 GB RAM as well, okay? So SAM and RAM, okay? So shoot apical meristem is SAM and root apical meristem is RAM. In permanent tissue, we have everything like we can say that parenchyma, sclerenchyma, collenchyma, xylem, phloem, everything will come into permanent tissue. All these things, okay? All of these things, they can come over here, okay? They can come over here, all right? So because all these tissues are permanent tissues, okay? So I can say that all these tissues have stopped dividing. Parenchyma, sclerenchyma, collenchyma, xylem and phloem have stopped the division process, so they can be put into this category as a whole, okay? So these are permanent tissues, and this is what is given in your NCERT, but it is not been explained very properly, as I believe, all right? So what do we have to do here? We have to study these things, okay? So let's begin, okay? So I'll just start the slideshow. All right, and okay. So what do we mean by plant anatomy? We mean by plant anatomy means study of tissue and cell structure of plants. In zoology, anatomy means study of the internal organs. So when we will talk about the animal anatomy, we will talk about the structure, the arrangement of tissues in their organs or in their cross section, etc. Okay, and this study of tissues is given a separate name. It is called histology. Okay, histology. Anyway, why do we call the cells so much important? There is a cell theory that all the living beings. It's not actually important. I'm just included it here. Cell theory says that all living organisms, whether that be a small organism, whether that be a large organism, small organism will be composed of less number of cells, large organism will be composed of large number of cells. So in cell theory, I've just written some things that all life is composed of one or more cells. Cells arise from pre-existing cells via cell division, okay? And sometimes the cells can fuse also. And cell contains DNA, etc. You already know it. Just the diagram of cell. It's nothing special. Okay. All right. What I want to show you here. Oh, sorry. What I want to show you here. Cell contains a large number of components, and these components can be the components that you see here. And please note that there are some extra things also. These are pigments, acids, tannins, gums, etc. Okay, a lot of things can be found in the cell besides the cell organelles. All right, next the slide. These are ergastic substances. These are also present in the cell. We will talk about them later at the end of this chapter. All right. Okay. Just take a quick look. This you do not need to write this all down. Okay. All right. Yeah. I have included this diagram because it means something. There is something you need to know. Okay. Why I have included this? Because every plant cell, it is joined to the other cell with the help of something. Okay. You can suppose that there is a cementing material which is present between the two cells, and this cementing material, it is called the middle lamella. Okay. So any kind of cell, if it is accompanied by an adjacent cell, there will be always some cementing material present between these two cells. Okay. You can suppose this as you have seen the bricks. Okay. Bricks are joined together with the help of cement. So this cement, in the case of plants, it is called middle lamella. All right. Where do you see middle lamella around yourself? Do you eat middle lamella as well? Is there any sort of food where we eat middle lamella? Do you know any such thing? Come on, students. I am expecting an answer from you on this one. Middle lamella is the cement of the plant cell. Let me help you with this. You have seen bananas, you have seen apples and all these things. So let me tell you, when a fruit is ripening, okay, when a fruit is ripening, it starts to get softer, right? It starts to get softer. So as it gets softer, you can see that what is the reason why it is becoming soft? It has not become soft because the cells are dying or degenerating. That is not the reason. The reason is that there is a cementing material and it has turned from solid to fluid state. Okay, it has turned into a liquefying material, a liquefied material, and that is why it has become so loose. All right. So middle lamella you can see in jams, jellies. Okay, the cell is very hard. The plant cell is very hard. It has a cell wall around itself. So on the fruit ripening, it does not mean that the cells have started to liquefy. It is the middle lamella which liquefies. So any kind of tissue will have a large amount of middle lamella between the cells. Okay, I've just included this slide just to tell you about this, because it's very important that you know that any cell will be joined to another cell with the help of middle lamella. And another important thing before we move on to the tissues, suppose there is there are two rooms, okay, and both the rooms have a wall between them. Okay, so cells also are similar to these two rooms. Okay, and the important thing to remember here is that a cell has to communicate with the adjacent neighboring cell. All right. So how does it perform this communication? There is a thing here called, please see the slide here. There is a thing called, in the diagram you can see a word written, plasmodesmata. All right. Plasmodesmata is the protoplasmic connection between the two cells. Okay. Suppose this is one cell, this is another cell. This is one cell, this is another cell. Between these two cells, there is a aperture which joins these two cells. There is a hole in the cell wall which joins these two cells together, and here the protoplasm or the cytoplasm is joined. Okay, so this is called the plasmodesmata. Okay, yeah. So is all students, you are saying that you have 12 students. All right, government music school students. It would be better if you write your names as well. I need to know you as well. Okay, so write your names. Okay, I'll recognize the ones who talk to me. All right. Okay, let's continue. So you see this diagram. Okay, cell number 1, cell number 2, they have middle lamella between them. They have cell wall just sitting very close, tight to each other. And inside the cell wall, what will be present? The cell membrane or the plasma membrane will be present. Okay, all right. So let's continue. Okay, so this part is clear. Now I told you that the cells can be classified into two categories: meristematic cells and the permanent cells. Meristematic cells means the cells which are in the continuous state of cell division. And I told you that the meristematic cells means it can be shoot apical meristem or the root apical meristem. So what we have here is the shoot apical meristem, and shoot apical meristem we can see here. It is actually of many categories. We have shoot apical, root apical, and besides these two meristem, we have another category besides the apical meristem. We have another category called the lateral meristem, and there is a third category also called the intercalary meristem. I have not included it in my notes today, but I will just include that as well. Okay, that part has been missed out, but I have to complete these slides and then I'll show you it. Okay, so apical meristem is shoot apical meristem. Where will it be present and how you can see it? The easiest way to see these things is you take an onion, buy an onion from the market. Of course, you will not buy a single onion. You will take 1 kg or 2 kg and take this onion and put it in a bowl of water. Okay, put it in a bowl of water for a day or two with the root part in the bottom side. Okay, so when you put this onion in the bottom side, maybe you have performed this experiment already in class 9th and 10th. If you have not done it, it's very easy. You can do it at home. Take an onion, put it in a bowl of water. Within, let's say about 6 hours, you just go to sleep, and after you wake up tomorrow, you can see that there will be the development of roots rapidly from that bottom part. Okay, so why has there been a development of roots in that region? Are you clear on this? Hello, Itanagar students. You are wishing me good afternoon when it's nearly time for good evening, right? Anyway, good afternoon to you as well. All right. Itanagar, please report your attendance. You have missed some things, I believe. I'll include my notes. I'll send them to you later. All right. So let's continue. You have missed quite a things, Itanagar. I was talking about the tissues. I have already talked about the kinds of tissues and the kind of meristems here. Anyway, apical meristem, shoot apical meristem, root apical meristem. Shoot apical meristem is a bit difficult to observe. You will need spatial arrangement. You will have to cut the apical meristem, shoot apical meristem at the precise point and then observe with the help of a microscope. But root apical meristem, we have quite a lot of examples we can see easily. You can take garlic, you can take onion. Onion shows a more rapid growth, so it's the best form of material to do this sort of thing. Okay. All right. Hello, Ramengi. I recognize you. And Himachuani, Pratima. Yeah, Pratima, I recognize you. Chami, Kori, Moanpoi, Sankti, Saga, Liansanga, and Esther. Yeah, I'll send them to you. Yeah, I will do that. I'll do that. You are a bit late, so please catch up. Okay. At least open your NCERT. That will be a little bit helpful, but this diagram is not there in the NCERT. Okay. So please try to follow up. Yes, Itanagar, I saw your chat. I'll send it. I'll send it. Yeah, great. Okay, so please do not spam so much. I understand. All right. Hello, Savina. Okay, so I'll just send them to you. Okay. Continuing. Root apical meristem is a bit easy to observe, I said. And if you have a microscope in your school, even a low power microscope will do. Take these newly formed roots and take a cross section and put them on a glass slide and cover them with a cover slip and observe them on a low power microscope or a high power microscope. You can very easily see the state of cells. Okay, they're in a rapidly dividing state, and you can maybe, if you are lucky, you can even see two cells which have just started to divide or have completed division. If they have completed the nuclear division, you can see in a single cell, you might be able to see two nucleus within a single cell. That means they have just completed the cell division. Okay. No problem, Itanagar. It's no problem. I'll send them the notes to you. Just try to cover up, okay? So this is about root apical meristem, RAM, and then let's talk about SAM, shoot apical meristem. Shoot apical meristem you can see in any plant on the growing tip part. On the growing tip, you just need to take a sharp blade and perform a cut, and these cells will be green in color, unlike the root apical meristem which were white in color. You already know that root is non-green achlorophyllous part, so it will not be green in color. But the shoot apical meristem, it might have a slight green color, okay, very slight green color. But if you observe it into the microscope, you will not be able to observe any chloroplasts. Okay, I'll explain why. You will not be able to see any chloroplasts because apical meristem is in a state of continuous division, and these cells are undifferentiated cells. Undifferentiated cells means these cells have started to divide, but they have not completed a sort of maturing process. Okay, so let's compare them to children. Okay, so they have not chosen their career, whether they will be part of the root, stem, leaf, or anything, whether they will be part of the flower. Okay, so they have not chosen their career. These cells are still in class 10th, right? So they have not chosen whether they will take biology or they will take maths. So about the lateral meristem. Lateral meristem is very important. Lateral meristem means the vascular cambium and the cork cambium. Vascular cambium means you have seen plants that they are very, very thin at the initial stage. Woody plants. In northeast you have a large population of teak trees, right? And teak trees have a very thin stem when they are growing up. They have large, broad leaves. Okay, Itanagar, it's perfectly okay. I'm not angry with you. Okay, I'm just saying that you have to cover a bit. Do not worry. You can always ask me. It's okay. All right, no problem. All right. So let me continue. So what happens is, in the case of teak plants, they are very thin. You have seen the teak plants around you. I know that northeast has a very good population of teak plants, teak trees. They grow in abundance there. So teak trees are very thin in the initial stage, but as they complete every year, they start to grow thick rapidly. Okay, so they gain a diameter very rapidly. Okay, so why does that happen? Why is that plant thin in the initial stage but becomes rapidly so it rapidly increases in diameter? The reason is it has a lateral meristem. Okay, it has a lateral meristem which helps it grow on the diameter side. Okay, it helps in growing it towards the diameter side. So it's a diametric growth. It's a diametric growth. And in the case of apical meristem, what was happening? It was increase in the length. This is an increase in the girth, the thickness part. Okay, so lateral meristem, apical meristem, and there's a third meristem which is a bit less important from the exam point of view. It is called the intercalary meristem. Intercalary meristem is not found in every plant. It is usually found in grasses and monocots. Okay, so I'll explain why. Intercalary meristem is present in grasses. You have a school ground, you have a playground, and you people play football, right? So just before any particular match, just before that, the ground has to be cleared out. Okay, it has to be cleared out. The grass has to be rapidly cut off. Okay, so we have a machine which rapidly cuts the grass. Okay, so the grass blade has reduced in size. Okay, and just after a day or two, you can see the grass is growing rapidly. All right, you have all seen this. If you cut the grass, or just imagine a green pasture in which the sheep or the herbivores are grazing, even after they have grazed off, or you have cut the grass with a lawnmower, after that the grass rapidly increases in size, and within 2-3 days or within a week, it will be on its previous size. It will gain the previous size. Why does this happen? We have cut the top part, we have cut the apical meristem of the grass. So why has it increased so rapidly in size? There must be some sort of a meristem. Sir, do a single plant contain all the three meristem? Yes, a single plant can contain all the three meristem. In the case of grass, we have apical meristem, root meristem, as well as lateral meristem. Okay, so we have apical meristem as well as intercalary meristem, but we do not have a lateral meristem. But in case of some plants, it might be possible that they have three types of meristem at the same place. But usually a plant will have either apical and lateral, or apical and intercalary. Monocots have apical meristem and intercalary. Dicots have apical meristem and lateral meristem. Okay, so and not also like that. Not all monocots have intercalary meristem. Okay, if you cut a wheat plant or a maize plant, it won't be able to increase in size so rapidly as compared to the grass. Okay, why? It has a different function. Look, monocots are short plants. They have a short size. So monocots, if we cut them off, they need to increase in size rapidly so they can perform photosynthesis. All right. They do not have a large number of buds. In case of woody trees, what happens is, if you cut a tree from the ground at about 2 feet or 1.5 meters from the ground, there are some dormant buds on that woody part, and after let's say about a month or two, you can see there are new shoots, new branches arising from that cut off woody tree. Okay, so why is this happening? There are some dormant buds in that trees. Every tree has that. Every dicot tree has that. But in case of monocots, they have only the help of intercalary meristem. They do not have so much buds. They have buds, but not as numerous as dicots. So every plant is different. They have some nature. We have to study this nature of the plant. That's what biology is about. Okay. So there is not a particular concept why this happens. But the thing is, every plant has to survive, and in order to survive, if we cut off the top plant, it has to show some sort of adaptation. So monocots adapt by rapidly increasing in length. The dicot plant has a large storehouse of food in the case of root. So dicots have a large root system. Monocots have a short, weak root system. So the dicot plant has already stored a lot of food. So it does not have a need or not an immediate requirement that it should increase in length rapidly. It has a large storehouse of food. So the buds will grow out in about a month. Okay, it's a slow process. But in case of monocots, if it does not increase in size, there is no storage organ in like the roots store of food, but that food is very less. So it has to increase in size. It has to perform photosynthesis. Otherwise, the plant will die. Okay. Why this plant is so differentiated? Itanagar students, you are asking me why these plants are so differentiated characteristics in the same place. I do not understand your question, Itanagar. Please repeat your question. I am not able to understand. Okay. All right. So one more thing. Where are the apical meristem tissue here? The apical meristem tissue is in the top. Okay. And in case of the diagram A that I am showing here, it is case of shoot apical meristem. Root apical meristem, it is a bit different. Root apical meristem is protected by some region, and this region is called the root cap. Why root cap is present in the case of roots? Because in the case of shoot apical meristem, there is no resistance to the growth. There is no resistance to the growth. It can grow indefinitely. But in case of root apical meristem, there is a resistance to the growth. Why? Because there are soil particles. If a cell is rapidly growing in length, it will get damaged by the root, it will get damaged by the soil particles. So it needs a helmet, it needs a protective device. So this helmet is called the root cap. So you can say that in case of shoot apical meristem, the meristem is terminal in position. In case of root, it will be subterminal in position. Okay, subterminal means next to terminal. So you can see the difference in arrows here. Please see this slide. There is a difference in the arrows here. There is a difference in the arrows here. Shoot apical meristem on the terminal part, root apical meristem slightly above the apex. All right. So there is a difference here. Okay. All right. So tissues and all these tissue systems are also classified into various categories. One category, one parameter is dermal vascular ground. Okay. And there is another also. Just see this. I'll give these notes to you. Okay. Ground tissue, parenchyma, collenchyma, sclerenchyma. Let's complete this first. Okay. So where have these tissues been formed from? From the meristematic part. The shoot apical meristem has undergone cell division, and after undergoing cell division, these cells, they have differentiated, they have chosen their career path. They have chosen to become parenchyma, they have chosen to become collenchyma, or they have chosen to become sclerenchyma cells, or they have chosen to become xylem fiber, or they have chosen to become phloem fiber. All right, all these kind of cells. So let's see the permanent tissue kind of cells. Permanent tissue kind of cells, the first cell is the simplest cell called the parenchyma. So where can you see these parenchyma cells? Where have you seen these parenchyma cells? Just take a potato, okay? Cut a potato, the mass of potato, the slice of potato that you are seeing, it is nothing but parenchyma cell, a modified parenchyma cell which has accustomed itself to storing a large amount of starch. Okay, so parenchyma cell is an undifferentiated cell. The most basic function of parenchyma cell, it is to transform into any other kind of cell if the need arises. Okay, and otherwise it will function as a filler kind of cell. It will function as a sort of cell which stores starch and helps the other cells. It's a supporting cell. Okay, so how are parenchyma cells looking like? They look isodiametric, or they may be elongated, and they have a very simple cell wall structure. They do not have a very thick cell wall. One more thing, students, you need to know here. You already know that cell wall is present just outside the cell membrane. The first cell wall that develops, it is called the primary cell wall. After that, the next cell wall that will develop, it will be the secondary cell wall. Then will be the tertiary cell wall. So a parenchyma cell just has, it only has a primary cell wall. Okay, it will have only a primary cell wall. But as a cell has the necessity, the requirement to increase in the thickness, increase in the strength to provide mechanical strength, it has to increase the size of its cell wall. If it does not increase the size of its cell wall, it will not be able to provide the mechanical strength. Okay. So I have a question for you. Please tell me, primary cell wall is the first cell wall. Now you have to tell me the secondary cell wall, where will the position of secondary cell wall be? You have two options. Primary cell wall, you already know, it is the first cell wall. Now tell me the position of secondary cell wall with respect to the cell membrane and primary cell wall. You have two options. First option is option A, the primary cell wall, the secondary cell wall, the next cell wall that will develop, the secondary cell wall is placed outside the primary cell wall. Okay, that is, first comes plasma membrane, then comes primary cell wall, and then comes secondary cell wall. The second option is secondary cell wall is present between the primary cell wall and the cell membrane. So please answer me option A or option B. Please give it a thought. I'm waiting for your answers. Please apply some logic to it. Okay, this question is based on logic. Whether the secondary cell wall is present between plasma membrane and primary cell wall, or is it present outside the primary cell wall? Option A is outside the primary cell wall. Option B is between primary cell wall and plasma membrane. So please, as all Itanagar, all students, please answer. I'm waiting for your answers. A or B? I believe you have heard my question well. Do I need to write it? Outside, Lunglai students, you are saying outside next to primary cell wall. Alright? Yeah? Okay. Meso school, outside the primary cell wall. Okay. All the other students, please answer. I do not believe you have a single answer. Okay, so Itanagar students, between cell wall and primary cell wall. Whoever is answering the question, please write your name in brackets after the answer. Just write your name after the answer. Okay, don't worry, I'm not going to scold you all. Okay, so just write your name. I just need to know your names as well. So Lunglai students, whoever has been saying that outside next to primary cell wall, just write your names. And okay, is all, yeah. Pratima, you are saying that outside the primary cell wall. Okay. And okay, Warrior, you are saying that, Warrior, you are saying between plasma membrane and cell wall. Okay. Other students, please answer. Please hurry up. It's a conceptual question. Please do not waste so much time on it. Option A or B, you can just answer option A or B. Okay. Lunglai. Okay. Let me be clear on this. Just please try to understand. A cell does not have its hands or something. It does not have legs. It does not have hands. Everything is supplied to the cell in the form of small molecules. These small molecules cross the cell wall. They cross the cell membrane. Okay. So if a cell has to make something, it will only be able to deposit it on the inside. Suppose this is a room and I'm sitting inside here. I have to make my walls thicker. There are some zombies coming. So all of you are playing PUBG, I suppose, okay? And there are some other games as well. So you are playing some zombie apocalypse something, and you need to strengthen your walls, okay? You have seen I Am Legend and a lot of other movies. So you need to strengthen your walls or the zombies will come inside. So what will you do? You will reinforce your walls. But you cannot go outside, okay? You have to reinforce them from the inside. So a secondary cell wall will always be present between the plasma membrane and the primary cell wall. It will not be present outside the primary cell wall. You cannot go outside. The cell does not have hands or anything so that it will deposit it on the outside. It will be deposited inside. And then what about the tertiary cell wall? Where will the tertiary cell wall be? Okay, Swagata. Yeah, Swagata, I recognize you. Very good. Swagata and Pratima. Yeah, very good. Saini also. Yeah, good. Yeah, Warrior also. Okay. So now tell me the position of the tertiary cell wall. You need to understand this because we are studying tissues, and then you will be able to understand this concept that why some cells become dead after the cell wall deposition. Please tell me the position of tertiary cell wall. I hope it is clear to you. Is tertiary cell wall present outside the secondary cell wall, or is it present between secondary and the plasma membrane? You have heard my question? You have heard me, right? So tertiary cell wall position, is it present between secondary and plasma membrane, option A, or is it present outside the primary and secondary cell wall? Yeah, I hope it is clear. It is going to be present inside the secondary cell wall and between the plasma membrane. So now try to understand this. There is a parenchyma cell. It is the simplest cell. It is the first cell. Okay. So this parenchyma cell, it has a very thin primary cell wall. Now, as the cell has decided to increase its thickness, it will develop a secondary cell wall, and then it develops a tertiary cell wall. Primary, secondary, tertiary. After the development of tertiary cell wall, any cell, it will become dead. Why it will die? Because a cell obtains the substances from the outside. These substances have to cross the diffusion barrier, and diffusion is proportional to the it's inversely proportional to the thickness of the membrane. Okay, you already know it. Thinner the membrane, higher the diffusion. Thicker the membrane, the diffusion will be proportionately very less. So any cell which has a very thick cell wall, it will always be dead. Okay, example is sclerenchyma. Example will be xylem vessels, xylem tracheids, xylem fibers, and phloem fibers. Okay, so another thing is the lumen, the inside diameter, the living space of the cell, it will be also very reduced. So because of these reasons, any cell having a secondary cell wall as well as tertiary cell wall, it will definitely be dead. Okay. So at the beginning, I told you that there are some plasmodesmata and everything. So parenchyma will have very clear-cut plasmodesmata connections between cells. Okay. All right. So this is clear. Parenchyma, then comes collenchyma. Collenchyma, you just see this diagram. In collenchyma, you can see that the cells have a very thick cell wall, and besides this thick cell wall, it has a very dense middle lamella. Okay, it has a very dense middle lamella. This middle lamella, this cementing material, it is holding the cells very tightly together. What is the function of collenchyma? The basic function of collenchyma is that in the beginning stage of the plant, development, the problem is it does not have wood. It does not have a support of a wood. Wood does not develop in the initial stage. So who is going to provide the mechanical support to the growing parts? It will be collenchyma. All right. So a very thick cell wall. Okay. Primary cell wall is very thick. It does not have secondary and tertiary. Please remember this. Collenchyma is living tissue, so it does not have secondary, it does not have tertiary cell wall, but the breadth of, the thickness of the primary cell wall is very high. And then there are middle lamella, it is also very dense. So parenchyma is living, collenchyma is living. Let's move on to the dead tissue. Okay. So these were at some point living tissues, then they become dead. So welcome to living dead. A very pure PG, I believe. Yeah. So what we have here, just a second. Okay. So please see this diagram carefully. Diagram A shows a very elongated pen-shaped cell, a very thin cell, and diagram B, it's a cross section. Okay. So a cross section shows the section which has been, suppose this is a cell which I am trying to represent to you here. This is a sclerenchyma cell. It is called a sclereid. Sclerenchyma has a great diversity. There are elongated cells. There are rounded spherical cells. There are star-shaped cells. But the special feature of all these cells is they have a very thick cell wall, primary, secondary, and maybe even tertiary cell wall. The important thing to note here is that the primary cell wall is thick, then secondary cell wall is very thick, and then the living space of the cell, it has become too congested, the cell dies, and the main function of it will be mechanical support. So where can you see this around yourself? Okay, where can you see this? I believe it must be in your room somewhere or outside your room. Can you search for the sclerenchyma? Can you search for sclerenchyma in your room? If there is a broom lying nearby, I'm not asking you to take out the table on which you are sitting, table and chair. I'm not asking for that. That is xylem. Okay, it is not sclerenchyma. I'm talking about sclerenchyma. It means the cells present for mechanical strength in monocots. So if you have a broom lying nearby, take a piece of it and cut a cross section, you will see sclerids and stone cells. Okay, so these are the elongated cells which are forming the part of mechanical support in many plants. Okay, so secondary cell wall and primary cell wall, dead end maturity. I hope this is clear to you. And you must also have eaten these pears. Okay, so a pear has a very gritty taste. Okay, you try to eat it and there is a distinct flavor to it. Guava, pear, and chiku, Acrus sapota. I am not sure what you call it. I call it sapota. The botanical name of chiku is Acrus sapota, Sapotaceae family. So you have eaten these fruits, and when you try to eat them, you have noticed a gritty taste. Okay, something is there which distinguishes it from mango or apple. Apple has a very smooth taste. There are no stone cells on that. But in case of pear, in case of Acrus sapota, all these fruits, they have some sclerenchyma cells, round sclerenchyma cells. These are called stone cells. Now sclerids are elongated, stone cells are rounded, they are spherical. Okay, so I'll discuss that also later. Please see the diagram of the stone cells. Okay, I have one in my home. Yeah, great. Just soak it in water. To observe the sclerenchyma, the sclerids, you take a broom, take a single part of that. Okay, take it out, take a single fiber of that, soak it in water and mix a small amount of vinegar and leave it for 2-3 days. It will become softer, it will become easier to cut. Or you do not use acetic acid, just soak it in water and take a blade and cut the cross sections. Okay, so that way you can do it. Anyway, we have the stone cells here. See the diagram here. Sclerenchyma has sclerids, it has stone cells. The diagram of stone cells is shown here. Okay, and diagram D is the diagrammatic representation, and E is how it looks in actuality. E is the diagram, is the actual microscopic image of a cell taken from this pear. Okay, a pear fruit sap. It has been put on a glass slide, and we are seeing this. Okay, so another important thing, please note here. I had in the beginning of this class, I told you about plasmodesmata. Plasmodesmata were living connections between the two cells. So in case of plasmodesmata, they were forming a sort of living connection. So in case of the cells of sclerenchyma, please note here, there is something called pit. Okay, you can see here in this diagram, there is something called written as a pit. Okay, pit is the place where the plasmodesmata were once present. Okay, or where the plasmodesmata, the protoplasm is there the cell wall cannot deposit itself, and therefore it becomes a sort of clear pathway. Okay, in the dying stage of a cell, in the last stage of a cell, the cell tries to pump more materials to the inside to cover it, to make it as thick as possible. After that, the cell has to die, of course, but it will try to pump maximum materials inside. So plasmodesmata will help in doing this, and after the cell is dead, they will form a sort of pits. Okay, all right, so this is clear. Okay, Let's talk about parenchyma, sclerenchyma, collenchyma has been done. Now let's talk about the complex tissue or the vascular tissues. These are called the xylem and the phloem. Xylem means the water and the mineral conducting part, and it has two things. It has either tracheids and vessel, or in some plants tracheids may be present but vessels are absent. So you already know that in Bryophyta, Pteridophyta, in Bryophyta xylem is totally absent. In Pteridophyta and Gymnosperm, tracheids are present but vessels are absent. In Angiosperms, tracheids will be present and vessels will be present. Both will be present. But let's try to understand what is the difference between tracheids and vessel. Okay, so let me try to help you out with this. Okay, a tracheid is a very slender part. Okay, see this diagram. Tracheid is a very simple. A tracheid is a very thin structure, and it's in diagram A. Okay, diagram A, the portion which is on the leftmost part, that is a tracheid. Okay, and it's a very elongated, very thin structure. Okay, now let's come on to the vessels. Vessel, you can clearly see that the diameter of vessel is very much higher than the tracheids. Diameter of vessel is higher than the tracheids. So in case of tracheids, the vessel is the diameter is very less. Okay, vessel is having a larger diameter. So in which case do you think the water conduction will be more faster? In which case? This is a question. Please stop talking. I can guess you are talking a bit. Come on, please answer this quickly. Tracheid and vessel, in which case the conduction of water is going to be faster? Okay, please answer this quickly. Tracheids and vessels, in which case the water conduction is faster in tracheid, option A, or water conduction is faster in vessel, option B. Shillong student, vessels. Okay, just write your name also. Option B. Okay, all right. It will be better if you write your names as well. I will prefer it. Please try to develop this habit with me. We have to study for a long time. Just try to write your names as well, so that I can remember that you are committing some mistakes. Do not be afraid of mistakes. I used to commit a lot of mistakes also. Please do not worry about that. I'm not going to scold you. Nobody is going to say anything. Okay, yeah. So basically, some students are confused. Tracheids. Look, the tracheids are thinner. Okay, let me explain this to you. A tracheid is very thin. A tracheid is very thin, and it's a very defective sort of conduction pathway. Okay, all right, Padla and Apostle Paul. Yeah, great. So yeah, in vessels the conduction will be faster. In tracheids the conduction will be slower. Okay, a tracheid is something like this: a very thin, a very high, thick with cell wall, and very less lumen. Lumen means the internal diameter. The internal diameter is very thin. Okay, suppose this is a tracheid, and the lumen of the tracheid is only this much. Okay, so it's a very inefficient water conduction system. Also, tracheids are placed randomly. Suppose this is one tracheid, and there is second tracheid. These tracheids are going to communicate like this. There are some holes here. You can see in the diagram I showed you there were some pits. From the bottom part, the water is going to be sucked in like a straw. The water is going to come out from this part. Okay, and this tracheid is going to communicate with this tracheid in the form of sideways. And this is going to leak some water as well. The third tracheid will be placed on this side. The third tracheid will be placed on this side. So it's a very inefficient water conduction system. Compare this to vessel. This is a large diameter vessel. Okay, this is a vessel. It has a very large diameter. Okay, it has comparatively a thinner cell wall, and these vessels are placed on top of each other. Okay, they are placed on top of each other. Okay, so this is a continuous water flow system. It is a very efficient water flow conduction system. Yeah, Priyanka, Padla, and Paul, very good. You have correct answers. Okay, I remember that. So vessels is a very efficient water conduction system, and trachea is a less efficient water conduction system. So if you understand this, difficult to figure out of working of trachea. Okay, look, trachea is okay. Why the trachea is conducting water? Look, the thing is, trachea is placed close to each other. Tracheae are placed close to each other, but they are placed in a random sort of manner. Okay, they are not placed on top of each other. They are not placed on top of each other. They communicate. One trachea is going to communicate with another trachea by placing it along the side. Okay, if you are not able to understand this, just try to understand this. A trachea has a very less diameter. Internal diameter is very less. A vessel has a larger diameter. Okay, so a larger diameter cell is going to be definitely more efficient than a thinner diameter trachea. Okay, plants which have vessels, they will conduct the water rapidly. Plants which have tracheae only, they will conduct water slowly. This is the reason why gymnosperms like pine trees, cycas trees, they are less successful than angiosperms. Ferns and gymnosperms, they are found in very less number. Have you wondered why? The reason is gymnosperms are limited to very few regions of the world. They are found in cold climates only. Cycas is an exception, and then too, it is very slow growing. Gymnosperms are very slow growing. Pine tree is very slow growing tree. Compare that to a teak tree, shisham tree, any other tree. These trees are very fast growing trees. Why? Because angiosperms have vessels. They have very good water conduction pathway. If water is going to be conducted at a very fast speed, it means water will bring minerals with itself. Plant will have a faster rate of growth. This is why angiosperms are able to grow rapidly, faster. So gymnosperms will be left out of the competition. They will be very slow growing. Okay, this is why angiosperms are more successful, more in number than the gymnosperms. Okay, this is the reason. All right. So tracheae have pits only, and vessels have perforation. You can see there is very clear in the diagram here. Tracheae have no perforation. They have only pits, small apertures, small pits. Vessels have large perforation plate, a large diameter plate. Okay. Why is there a exceptional cycas in biology? I'll discuss that later maybe. Each another student, please write your name and just talk to me. I'll clear a doubt about that. Cycas spelling is wrong. Cycas. Meso school, you are asking me what is the difference between angiosperm and gymnosperm. Look, angiosperm has ovary. Angiosperm has ovary. Gymnosperm has naked ovary, naked ovule. No ovary is present. Okay. What is the difference between these two? I told you. In gymnosperm, the ovules are kept on the leaf like that. Okay. They are not covered. In angiosperm, it will be covered. How covering is formed? The ovary is formed by the protection of the ovule. Okay. The carpel, it folds itself. If the carpel is going to fold itself, the seeds will have a protective layer. This is called ovary. In gymnosperm, the ovule are present as such. They are kept on top of it like that only. Okay. Gymnosperm has no vessels. Angiosperm has vessels. Gymnosperm have cones only, they have no flowers. Okay, seeds are formed, but fruit is not formed. Angiosperm has fruit, it has seeds also, it has flower also. Okay, so many large differences are present. I am just telling you small number of differences. And Itanagar student, you are asking why is there an exception in cycas in biology? The thing is, cycas can grow in warm climate as well as cold climate. It has adapted itself to both places. I'll tell you why gymnosperms are present in cold climates only. In cold climates, angiosperms cannot grow well. In cold climates, angiosperms cannot grow well. Why? Reason is, angiosperms do not have, not all angiosperms have xerophytic adaptations. And even after xerophytic adaptations, they are, in case of xerophytic adaptation, like thick cuticles, sunken stomata, water conserving power, angiosperms are weaker than gymnosperms in this respect. Okay, so gymnosperms are slow growing. They are able to conduct water through their stem less efficiently, but they are very good at conserving water. They are very good at conserving water. Angiosperms are poor at that. So in the cold regions where the availability of water is very less, in that case, gymnosperms are successful than angiosperms in that condition. So that is why in the case of mountain tops, you can see pine trees only or devdar trees only. You will not find angiosperms, you will not find teak plants, you will not find mango plants, you will not find neem plants, okay? Because angiosperms are not successful over there, okay? That is the reason. Exception, there is an exception in gymnosperm also. Neetum, ephedra, welwechia. 3 plants have primitive vessels, but I will not discuss that here. Biology is the science of exceptions, okay? Later, I'll tell you which gymnosperms have primitive vessels, exceptions. Alright, let's move on to phloem. Phloem has sieve cells and companion cell. Phloem is form of phloem fiber, phloem parenchyma, phloem, sieve cell or sieve element, and next is the companion cell. A question is asked in your exams that which cell in plants does not have a nucleus, but it is still considered as living cell. Do you know the answer? Which cell does not have a nucleus, but it can remain living for a long amount of time? It is a living cell. It will not die. It will not die quickly, I should say. What is the example of such cell in plant kingdom? Do you know the answer? Come on, students, please answer quickly. We do not have much time left. Or do we have some, I'll have to see this. Okay, so please answer quickly. You do not know the answer, or you do want me to answer? Which cell, I'm asking you. I am asking you. I do not know the answer. Seni. Lungla students, I do not understand your answer. The answer is sieve cell. The sieve cell, it does not have a nucleus, but it is still considered as living cell. Okay? The sieve cell, it is considered as living cell. Okay? The sieve cell does not have a nucleus. In the case of animals, the cell is called the RBC. It does not have a nucleus, but it has a living thing. Okay? So, please try to understand this. Alright. So, let's continue here. Okay. Alright. Sieve cell, it does not have a nucleus, but how does it remain living? It has a cell called the companion cell. The companion cell sits close to the sieve cell. The companion cell sits close to the sieve cell, and in case of companion cell, it supports the sieve cell and keeps it in a living condition. All right. So a sieve cell has a function of performing the food conduction pathway, and by the food conduction pathway, I mean the flowing of the sugars, the transport of the sugars from one part of plant to the other plant, to the other part. Okay. So you can see the diagrammatic representation, and on the right side, on the B side, in the C side, you can see the actual appearance under the microscope. Okay. The A is the diagrammatic representation that you have to draw in the exams, and in B and C it is the how they are observed. Okay. All right. So this is the actual microscopic diagram. Okay. Why is the nucleus important for the cell? Is all students, you have asked a very important question from me. Why is the nucleus important from the cell? So look, nucleus can be said to the brain of the cell. Okay. Whatever the function the cell is performing, everything is controlled by the nucleus. Okay. If the nucleus dies, the cell will die. Nucleus maintains the cell. It repairs the cell. Okay. It keeps the cell in proper order. Okay. If the nucleus is removed, the cell will die. Okay. That is the thing. Nucleus has DNA. DNA forms RNA. RNA forms proteins. RNA forms enzymes. Okay. So everything, for maintaining everything, you need the nucleus. Okay. That is the reason. All right. So let's move on. Okay. This is actually not so much important. Yeah. Okay. So now I have to discuss something called the root region. Okay. The root region, I have to discuss this. Look, we have to discuss the root region. We have to discuss the shoot region also. In case of root, the region, if we cut a longitudinal section, you can see, have you ever pulled out a plant from the potted condition or from the field? When you put out a plant, take out it, uproot the plant, what will happen is, when you uproot it, you can see there is a network of root hairs. Okay, when you uproot the plant, you can see a network of root hairs. But when you see at the bottommost part, you can see there is absence of root hairs. Okay, there is absence of root hairs. Now let me tell you again. I told you about root apical meristem, the ram. Okay, root apical meristem is covered by helmet-like structure. It is called the root cap. It is shown in this diagram here. Above the root apical meristem, the cells which have just been formed, newly formed, they are present. This is called the root elongation zone. Okay, root elongation zone. Root elongation zone means those cells which are just formed and they are going rapid elongation. Okay, rapid elongation. So these cells are increasing in length. These cells are increasing in length. Okay, and above the cells, above in the topmost part of this diagram, you can see root hair zone or the root maturation zone. So 4 zones are present: root apical meristem, root cap, root elongation zone, and the root maturation zone. Okay, the meristematic zone, root cap, and the elongation zone and the maturation zone. Elongated cells, after they have become fully mature, they will start developing root hair or they will become permanent cells. Okay, all right, let's move on. This is a transverse section through a root. Okay, transverse section through a root. All right. Okay, so there is something called a Casparian strip. I need to explain this. Here in this diagram, you can see something here. If you cut a cross section through a root, you can see that there are some cells present which have developed cork inside the cell. Okay, A cork is a fatty substance which protects the cell from bacteria. It does not undergo degradation, and it is waterproof. Okay, so inside the root, why do you think that some sort of cell will be present which is having a waterproof property? Okay, Government Mizo School, you have a question from me. You have a question for me. Sir, red blood cell do not have a nucleus. How its function is controlled? Yeah, very good question. Let me answer it. The thing is, a red blood cell has developed the property of losing the nucleus. A red blood cell, in our case, in humans, it is devoid of nucleus. But in case of amphibians like frog, lizard, their cells have they do also have a red blood cell. Okay, they have a red blood cell, and their red blood cells have nucleus. They have nucleus. We do not have a nucleus. So, do you think it's an advanced feature or a let's say a reduced feature? Is it a primitive feature or a advanced feature? We are humans. We are definitely more advanced than amphibians. Okay. So the thing is, red blood cells nucleus lose the nucleus so that they can store more hemoglobin. Okay. They lose the nucleus so that they can store more hemoglobin. But your question is, why does it not have a nucleus and how its function is controlled? I have cleared the first part of your doubt. It does not have a nucleus because it wants to store more hemoglobin. It becomes more efficient. And how its function is controlled? Its function is controlled for a limited period of time. It cannot control its functions. The enzymes that are present inside the red blood cell, they will function on their own. Okay, and after some time this cell is going to die. Red blood cells have a lifetime of 100 days or 120 days. As soon as they have become old, they have become functionless. Our spleen is an organ, and spleen filters these old cells. It destroys the old cells. Okay, and then new cells will be formed. So continuously new cells have to be formed, old cells have to be destroyed. So red blood cells do not have a permanent lifespan. They cannot live indefinitely. That is the reason. Okay. So my question to you was, why is a waterproof substance? The root has a function of conducting water. Why do we have a waterproof substance inside the root? Why do we have a waterproof substance inside the root? Do you know the reason? Okay, the reason is, I do not believe you will be able to answer it. The reason it will be more complex. Look, there is a root. Okay, imagine a circular cross section of the root. Okay, inside, and inside the circular cross section of the root, there is a region called endodermis. Okay, there is a region present called endodermis, and in endodermis, some cells have these red portions called cork coated portions, but in some portions, the Casparian strips are absent. Okay, Casparian strip, it will not be present in the form of complete circle. If complete circle is present, then the root is going to die, the plant is going to die. There will be no conduction of water. There are holes, there are apertures, there are holes where Casparian strip is absent. Now, why is this substance present? It helps in increasing the pressure of water. Okay, try to understand it like this. You have seen a water dam, a hydroelectric dam or a water dam. What happens in a dam? We collect a large amount of water and force the water to flow through a particular region, and below that we store a electric generator, a dynamo, okay, a turbine, and that rotates and performs motion, and that motion is converted into electrical energy. Similarly, this plant is trying to increase the pressure of the force of water flow inside the xylem. What is it going to do? It forms a barrier, and at some points the barrier will be open. Water will rush from all sides, but at these points the pressure, the force of water flow will be larger. That is why Casparian strips are present. Okay. All right. So let's move on. Okay. Now I already showed you the regions of the root apical meristem. Now let me show you the regions of shoot apical meristem. Okay. So how does a shoot apical meristem form new leaves? You can see here, apical meristem, it is forming leaf-like structure called the leaf primordia. In the left side, it is the diagrammatic representation. Right side is the actual figure, actual cross section of a shoot apical meristem of a plant. Okay. So below that there are green portions, but on the top of that you can see non-green apical meristem is present because it is undifferentiated part. All right. So leaf primordia are forming, they will be converted into new leaves, and then you can see bud meristem, dormant meristem. Whenever the plant is growing, it leaves some dormant meristem cells. Dormant meristem cells, these are called buds. Okay, what is the function of buds? Suppose we cut a plant, suppose a shoot apical meristem is cut away, then these dormant buds, they will become active and they will form the new branch, new stem. They will start growing upwards. That is the function of buds. So while the shoot apical meristem is growing upward, it will leave some cells which are meristematic cells, but they will not be dividing right now. Okay, they are dormant. These are called buds. Okay, coming back to the diagram here. All right, so buds are present here, shoot apical meristem. Okay, talking about stems now. Stem, you already know. Okay, what is that have? It has an epidermis outside, inside portion called cortex and all that. Xylem phloem also you can see here. Okay. All right. So steel I will maybe discuss, maybe not discuss. It will depend how quickly you people are grasping things. Only then I will explain this concept. Otherwise, it might become too much for you. Okay. So now let me explain to you the function of lateral meristem. Okay. Lateral meristem, as I already told you, it helps in increasing the diameter, the thickness of the plant. So please see here, what you are seeing inside is the ring of cambium. Ring of cambium. This ring of cambium, it will undergo cell division by the mitosis type cell division, and it will form, after cell division, it will form layers. Okay, and these layers are going to be pushed towards the outward side. Okay, so in the first diagram you can see very less layers are present. Second diagram you can see large number of layers are forming. Third diagram you can see, fourth, fifth, you can see easily that new layers are being formed and they are being pushed outwards. Okay, all right. Now secondary phloem and secondary xylem will be formed. I will explain what are these. Okay, okay. Okay. So yeah, now let's see this diagram in detail now. There is a cell called cambium. It is present in dicots only, not in monocots. Cambium is absent in monocots. It is only present in the vascular bundle in case of dicots only, never in monocots. This is the reason why wheat plants do not grow in thickness. If they had cambium, then after some point of growth, they will develop a large woody stem. But cambium is absent in monocots. So it will not have any effect or feature. Okay. Okay, so vascular cambium, secondary phloem, secondary xylem. I'll explain these in detail later. Okay. Now here is a very detailed diagram of secondary growth. I have to cover this, but I will see if I can cover it here today. I'm not sure. Okay, so study this diagram here carefully. Okay, so study this diagram carefully for a second. All right. So secondary phloem, secondary xylem. Okay. Springwood, summerwood. That I will explain later. What is the difference between springwood and summerwood? Okay. All right. So yeah, I have to discuss all this, but in due course of time. Okay. So let me wait a bit. I have to show you something else also. Okay. So this is another one. I thought of showing this to you. Okay, so just read these points quickly. Okay, meristematic is concerned with growth, epidermis is concerned with protection, parenchyma storage, sclerenchyma strength and support, vascular you already know, xylem phloem, and this is the arrangement of tissues. Okay, xylem and phloem are present inside. They are present in the form of a bundle called the vascular bundle. And ground tissue it is composed of parenchyma. I told you that parenchyma has a function of filler tissue. It has a function of okay. All right. So okay. So let me continue here. Okay. Now roots general functions you already know. Anchor the plant, bind the soil, stops the erosion. Okay, this is not necessary, just telling you about that. You already have covered this in morphology. I have included morphology and anatomy in a single PowerPoint. Okay. Tap roots you already know. Just see the diagram. This is just for quick revision. Okay. Tap roots you already know. Dicots have tap roots, okay, and they grow deep into the soil. All right. So one thing you can see here in this diagram here, in the case of tap root, you can see that the root elongation zone, you can identify the root hair zone, root elongation zone or the root maturation zone, then the root elongation zone. Root apical meristem will not be visible, root cap will not be visible, but you can see at the bottom most part, root hair are absent. Okay. So in this diagram, just see this. Okay. All right. Fibrous roots, they lie close to the soil, are present in the case of monocots. Here, secondary growth is absent. Okay. They will not grow thick like the case of dicot roots. Okay. All right. Adventitious root. Now there are three kinds of roots. I already told you that adventitious, fibrous, and the tap root. Adventitious root is found in monocots as well as dicots. Students have a lot of confusion on this. Somebody was asking me questions also. The thing is, adventitious root can develop from any part of the plant. Okay, any part of the plant which develops a root-like structure, it is called adventitious root. For example, money plant. You put a money plant in a pot full of water, it will start climbing upwards. Okay, so it will develop roots at that point of attachment to the wall. So it is also an example of adventitious root. Okay, aerial roots. In Sikkim, if you have seen, I'm not sure that you have seen orchids or not. Orchids are present in northeast in abundance. They are not found in anywhere else in India in so much abundance as northeast. they have some things called aerial roots. Okay, okay, all right. Okay, I'll continue. Root hair, these are root hairs. Okay, regions of the root tip. This was what I was talking to you about. Root cap, the protection zone, the helmet-like zone. Then we have the root apical meristem zone. Okay, the region of cell division. Then we have the region of elongation. Then we have the region of maturation. The root cap protects it. Meristem divides it. Elongation is the rapid increase in length. Okay, yeah. So root maturation is the root hair zone. So if a question is asked from you, in which out of the 4 zones, if an objective question is asked, out of the 4 zones, in which zone is the root hair present? So we will say region of maturation. In which zone, the question is changed, in which zone rapid increase in the most fastest increase in the length of the root takes place, then you will say region of elongation. Okay, region of elongation means cells become longer. Okay, they increase in length. Region of cell division means cell division means that the region will increase in size, but the increase in size remains more or less constant. Okay. For example, in case of root apical meristem, the size of root apical meristem is about 1 millimeter only, 1 millimeter or 0.1 millimeter. But in case of shoot apical meristem, the size is a bit larger. Shoot apical meristem may be 1 centimeter in length. Okay, but it's normally remains fixed. It will not increase in size. The maximum increase in length takes place in region of elongation. Okay, cells become longer rapidly. Okay, it is so. If a question is asked, which region is responsible most for the increase in length of root, you will not write region of cell division. You will say region of elongation, because region of cell division is indirectly responsible. It is forming the cells, but in region of elongation, the direct increase is taking place. Okay, root cap is the protection zone. Okay, root tip. This is an actual diagram on the left side. Okay, this is just to give you a better idea. Okay, how this process happens. In the center, in the right diagram, you can see in the center part a blue colored, a violet colored portion is present. Okay, which I am calling stele. Okay, it's a vascular part. It is responsible for the conduction of water and the minerals, and also for the transport of food. Okay, just a better diagram. Okay, so function of root tissues: epidermis, cortex, endodermis, pericycle, xylem, phloem, stele, pith, apical meristem, meristem. These are technical definitions. I thought that it would be better if I include all this, because definitions are quite much asked in your exams and all that. So included it here. Okay, epidermis. What is epidermis? What is cortex? What is endodermis? I have to cover this also, but in the due course of time. Okay, all right. So coming on to this, we have the dicot root, the cross section. Dicot root cross section means what a dicot root appears to us in like cross section. Okay, so there is now the portion which starts now is the comparative anatomy of dicot root, dicot stem, and the monocot root and the monocot stem. Okay, so this is a diagram of dicot root. You have to remember it. I cannot do anything about it. You will have to practice it for several times. You will have to draw in your books. This is not the diagram you have to draw. I'll give you a better diagram than this. Okay, yeah. And then we have a monocot root. Please try to remember that in monocot root and monocot stem, secondary growth will not take place. In dicot root and dicot stem, secondary growth will take place. Okay, right. This diagram is not clear, but what I want to show you here is a very important thing. Please look at the center. please look at the center, both at the right and the left diagrams. In the center, you can see there is a star-shaped structure. Okay, a star-shaped structure is present in the center. This is the vascular part. The xylem vessels and the phloem cells are arranged in a star-shaped pattern. It is a distinctive feature of dicot root. It is a distinctive feature of dicot root. In monocot root, this structure is not present. In monocot root, this part will be different. Just try to remember this for now. Okay. Okay, so before I was asking you all this, I had asked you a question that what is the function of cork in the root, okay? So I had explained to you that it is a waterproofing material, and at some points the cork is absent. So now see in this diagram, Casparian strip something is present here. The red part is the cork part, and you can see that there are openings here. Can you see them, students? It is very clear. Red colored part, and then there is a green cell, okay, which is completely having no cork, okay. That is the part where the water will rush in with pressure inside the root cortex, okay. So that is that, right. So steel, don't need to discuss this, okay. So I hope it is clear to you now, okay. We studied about plant anatomy, tissues, okay, the meristematic and permanent tissues, okay. So all these things had to we have studied some slides, okay. So it has become clearer that how you have to study all this. I'll give you these notes, okay. Do not worry about that. I'll give you these notes, okay. And okay, so this was, I believe there is, yeah, okay. Okay. So I'll begin from the dicot stem and monocot stem, okay. Stem has following functions: support, conduction, growth, storage, okay. Okay. Support means stem support the plant's leaves, flowers and fruits, okay. That means is the function of support. Then we have conduction. Conduction means like roots, the stem contains vascular tissue, xylem and phloem. Xylem conducts water minerals, phloem conducts the sugars. Okay, an important thing here, phloem conducts the sugars, okay. So what are the examples in which the sugars created during the photosynthesis, they are used, okay. So can you remember that? Do you remember that? Can you give me a very good example where the phloem conducting the sugars is being used for consumption or anything? All right. Do you know toddy palm, or coconut palm, or do you know about maple tree, maple syrup? Do you recognize any of these terms? Do you know about toddy, or do you know about coconut palm, or do you know about this thing called maple syrup? Taming Long. Hello sir, what's the topic of today? You are asking me now. We are studying the plant anatomy. We are studying plant anatomy today. Plant anatomy means, Taming Long students, I do not know why you lagging so much. We are studying plant anatomy today. Plant anatomy means the internal structure of stem, root, leaf, and then we have to compare monocot stem with dicot stem. We have to compare monocot root with the dicot root. We have to study what kind of tissues are present Anatomy is the arrangement of tissues. So we have to study about, first we have to study the simplest part, the parenchyma, collenchyma, sclerenchyma, xylem, phloem. Then how are these tissues arranged with respect to each other? Okay, so arranged with respect to each other means we have to cut the cross section. So when we cut the cross section, we will understand that yes, this is parenchyma, this is collenchyma, this is sclerenchyma, this is xylem, this is phloem. Okay. Okay, so you already know this, we have covered this. Okay, let me move further. Okay, wait a bit. This is from the previous slide. Okay. All right, so wait a second, I have to forward it a bit. Yeah, there we are. Okay, so a stem, when we talk about a stem, okay, so I will start to perform some cross sections. Okay, I will try to cut some cross sections, so you will be able to study how the tissues are arranged with respect to each other. Okay. Okay, coming along, students. Okay, you have a satellite problem, so you joined a bit late. Okay, no problem. Coming along, students, I will share a PowerPoint presentation with you in your group. Just add me to your group if you have not already. I'll share this slide with you so that you can have the notes. Okay, understand your problem. If you see these slides, you will be able to. I'll give you the notes. Okay, do not worry about that. Okay. So stem anatomy. Let's start from the outside. The outside part is called the bark. Okay, bark is composed of not vascular cambium. It has cork cambium. It has some second primary phloem. Okay, other cells are also present, and inside this secondary phloem, cambium, and then secondary phloem, secondary xylem, cambium, and then secondary phloem, etc. All these things are present. Okay, so we'll discuss this. Okay, now see this very beautiful diagram here. This is a stem of this is a mature plant. Okay, can you tell me how old this plant is? Can you tell me what is the age of this plant from which this cross section has been taken? Do you have any idea about it? Do you have any idea how much old? Yeah, it's at least 3 years old. Yeah, it's at least 3 years old. Okay, so you know it very well that we count the things called annual rings. Okay, and that gives us an idea why how many years old the plant is. Okay, so you can see this very beautiful diagram here. Very clear wood vessels are present here, and it has a very distinct pattern. Okay, so very easy to see. Okay, so we'll come back to this later. Okay, so some terms you need to know. Wood is basically xylem, and wood has spring wood, large thin-walled xylem cells produced in spring season. Produced in spring season means the favorable season, so more resources, more food. So this means the cells that are going to be produced at this time of year, they will be larger in diameter. And the cells which are going to be present, formed in the time of summer, they will be summer and autumn season, and even we sometimes include winter also. Not actually summer wood, we called it autumn wood. Okay, so at that time, autumn and winter, no sunlight is present. Summer is a bit wrong. Summer is in those countries where summer is very harsh. Okay, no water is present. In some countries, winters are more harsher than summers because there is no availability of water also, and the leaves are all gone. Okay, so at that unfavorable season time, the vessels that are going to be produced, they are going to be very thin diameter. Okay, so this thin diameter means if you are observing it from a large distance, we can say these cells will appear like a line. They will be shorter in diameter, and the thick vessels which are present in larger, let's see this, okay. Okay, so the thin diameter cells, they are forming the rings, okay, dense, dark color, because they have a very thin diameter. The cells which are formed in favorable season, they are larger in diameter. So dark and light, dark and light. This pattern is called a annual ring. A single annual ring consists of one dark pattern and one light portion. So that is called an annual ring. Okay. One growth ring represents the amount of springwood and summerwood that has occurred in a single growing season. Okay. So this is, I believe, 3-year-old woody dicot stem. It is very clear to you. Okay. Yeah. So let me move on further. I hope you have seen this. Okay. Nothing to note here. This will be already given to you. Another terminology that is asked in your exams is heartwood and sapwood. Okay. So after many years of secondary growth, what happens is, after the secondary growth, try to understand it like this. After the secondary growth is happening, there internal tissues of the plant, the internal cylinder of the plant, it is having a large amount of pressure from the outside. Okay, the internal cylinder of the plant, it has a large pressure from the outside. So a large pressure from the outside means the internal tissues are going to be compressed. Okay, they are going to be compressed. So if they are going to be compressed, so if, there is going to be a large amount of pressure on it. So the internal cylinder, it will sort of collapse and it will become more dense. So if you have seen a very old plant, a very old tree, the internal cylinder, it is usually having a darker color than the outer cylinder. Okay, so internal cylinder is darker colored because of development of tannins, because of compression, everything. Okay, but all these cells are dead cells. Okay, because they used to conduct water at some point, but because of outer pressure, they have died. Okay, so you understand this? All right, but it helps in strengthening the trunk of tree. Okay, all right. Sapwood is the outer cylinder that is actively conducting water. It is lighter in color and surrounds the heartwood. Okay, so in this diagram it will be clear to you now what is heartwood and what is sapwood. Okay, so you have all seen this. Okay, internal cylinder is always dark colored and outer cylinder is always lighter colored. So it is called heartwood and sapwood. Okay. Now a very interesting application for all of you. There is something called a maple syrup. I am not sure if you know about it, but in supermarkets and in Darjeeling, you must have seen maple trees are present. In supermarkets, the maple syrup is marketed from Assam and Darjeeling areas. Okay, I'll tell you what it is. Okay. So in late summer, early fall, starches are accumulated in the stem and root. Okay, so actually what happens is the plant is storing some storage food material for itself. All right, so it's storing some storage food material. So storage food material is always starch. Okay, but in the next year, in early spring, when the favorable season has come, and the temperature is good, the starch is rapidly converted, very fast conversion, into sugars, and these sugars are rapidly transported. Okay, in early spring when the temperature is very good, the sugar is transported to the growing regions for the growth of new leaves, new buds, new branches. So this sugar is transported to those areas. So what do? We ask the plant that nicely, please give us some of this sweet solution that you have, and the plant agrees, yes, that yes, you can install a bucket on me and take some of it, and it is for you and your family. All right. So the water filled sap, it is collected in small buckets. Okay, you can see how it is being done. Okay, a cut is made, and a tube like metal cylinder is there, and it stores the flowing sap into a small bucket, and this is boiled, the water is evaporated, and finally what remains is a thick sugary solution. It is called a chashni in Hindi, or you call it syrup. Okay, in English we call it syrup, in Hindi we call it chashni. Okay, so a very thick solution, and it's very sweet scented, it smells very nice. It's a natural syrup. Okay, you do not have to add sugar to it. So it is called a maple syrup. It's very good to taste. Maybe you should try it sometime if you have not already. And this is called the maple syrup. So it's so just my question to you here is, you already know which parts of the stem conduct water and minerals and which parts conduct food. So please tell me what is the portion that has been captured or injured to collect the sugar syrup. You just please tell me which tissue to prepare maple syrup, which part has to be punctured, which part has to be let's say hijacked to capture the sugar solution. Please, I'm waiting for your answers. Come on, students. After that, I will answer why I was asking you about coconut, palm, maple syrup, and all these things. Please answer quickly. Which tissue is targeted to collect maple syrup? Which tissue or which cell? Come on. Is it such a hard question? Is it such a hard question? Xylem. Write your name also. Government Meso School. Xylem. Okay. More answers? Anybody thinks anything else? Anybody thinks anything else? Tamenglong, Lunglai. Okay, Pratima, you are saying xylem is responsible. Xylem is being targeted to collect the sugar syrup. Okay. Anybody has a different answer? Tamenglong, Lunglai, Shillong. Come on, dear students. Itanagar. What's your response? Write your name and write your answer. Come on, I'm not going to scold you, yaar. Please, you are sweet people. You should answer. What's holding you back? Am I boring or what something? If I'm boring you, just let me know. I'll try to make this a bit more interesting. Just let me know what you feel, okay? Just let me know your feelings. Phloem, Shillong, okay? Any more answers? Anybody thinks epidermis or parenchyma, collenchyma, sclerenchyma, anything else? Xylem, phloem, you have 5 options. Only 2 students have answered so far. Why is it happening? This is a very interesting thing. Are you not going to answer? Alright, let me tell you. Phloem is being targeted here. Phloem is the part which will always conduct sugars. Phloem is the part which will always conduct sugars. Xylem is always going to conduct water and minerals. So xylem is not correct. We are going to target phloem only. Now let me tell you, I was talking to you about coconut palm and about toddy and all these things. There is a palm called toddy palm. If toddy palm is not present, we can use coconut palm. In coconut palm, you on the top portion of the coconut palm and on toddy palm, there is a stem. Stem part is modified, it bears flowers, and these flowers are later converted into fruits. Okay, so what people do is they climb on top of the coconut tree and the toddy palm tree. They climb on top of it, and they cut this inflorescence axis. Okay, this has a very rich supply of phloem, a very rich supply of phloem. Xylem also, but xylem is less developed. Phloem vessels are phloem companion cells and sieve tubes are present in high number. So this has a very rich supply of sugars. What people do, they collect this sugary solution from coconut tree or toddy palm tree or maple tree also, and they collect it and they use yeast and they prepare very good quality fermented beverage called wine. Okay, toddy palm is used to make toddy. It's a alcoholic beverage, and coconut palm is also used to make toddy palm like thing. Okay, it's called toddy also, or some people call it coconut toddy. And maple syrup, this can also be used to prepare anything which is sweet in nature, very sweet in taste, which has sugars. It can be used to make wine. All right, so this is about that. Okay, so let us come back to the notes part. All right, so I was talking to you about the plant anatomy. So let us quickly finish this. What we have done so far, we have learned about the meristematic zone. We have done about the permanent tissue. We have learned about shoot apical meristem, root apical meristem, and then we have another lateral meristem. Okay, we have another thing called lateral meristem. Shoot apical meristem. Now let's finish this quickly. Shoot apical meristem, root apical meristem, okay. So shoot apical meristem is responsible for increase in length of plant or shoot. Okay. Similarly, this is responsible for increase in length of root. Okay. And lateral meristem, this is responsible for increase in girth. Okay. This is responsible for increase in girth. So this is what we have already covered. Okay. We have already covered this. Now about shoot apical meristem, you need to write a few things maybe. If you want to write, you can. Otherwise, just try to pay attention here. Just try to listen. Shoot apical meristem. Okay. There are a few things you need to know. Shoot apical meristem is present on the topmost part. Okay. This is the shoot apical meristem. Now, shoot apical meristem, sometimes it leaves dormant parts. These are called buds. Okay, buds are meant for growth. Okay, they will be used to make new branches. Okay, buds. Okay, and shoot apical meristem, it is present at shoot apex. Shoot apex. Okay. Length of shoot apical meristem is more than root apical meristem. Okay. And it consists of rapidly dividing cells. It consists of rapidly dividing cells. Okay. Now you need to learn a few things about the properties of rapidly dividing cells. Okay. In rapidly dividing cells, we do not need primary cell wall. cell wall is thin. Cell wall is very thin. Only primary cell wall. Only primary cell wall, okay, is present. Okay, and after that, what we have here, only primary cell wall is present. Okay, and besides the primary cell wall, cell vacuole is either very inconspicuous, cell vacuole is absent, or very much reduced. Or very much reduced. Another important thing, plastids are absent. Plastids are absent. Plastids are absent means it will not have a green color. Okay, chloroplast, chromoplast, etc. absent. Chloroplast is absent. Chromoplast is absent. Chromoplast is absent. Okay. Government visa school, you are asking me, can we extract the alcohol from tree? If you are asking me, if you are trying to extract ethyl alcohol, you cannot. Ethyl alcohol is always to be prepared by fermentation. However, you can extract another alcohol, it is called methanol. You can extract methanol from trees, but methanol is highly inedible. You cannot extract, you can extract methanol, but it will not be worth drinking. You already know the effects of alcohol. Ethanol is the least toxic alcohol, and every other alcohol is toxic. Okay. So methanol, propanol, isopropanol, all these alcohols, they are poisonous, but you cannot extract ethyl alcohol from any tree. You have to extract the sugar solution, then you have to wait for the yeast to perform anaerobic respiration of fermentation, and then ethyl alcohol will be prepared. So you can extract maple syrup, sugar syrup in dilute form, but you cannot extract alcohol directly. Okay. So I was telling you, plastids are absent, chloroplast and chromoplast are absent. And another important thing, these cells are very small in size. Okay, cells are small in size. Okay, cells are small in size. Okay, so we learned that vacuole is absent. Okay, plastids are absent, cells are small in size, cell wall is thin, primary cell wall is only present, secondary cell wall absent, tertiary cell wall absent. Okay, yeah. Okay, so now tell me, if I'm telling you all this, what cell organelle will be responsible for the maximum increase in length of a cell or a diameter of a cell? Do you know about all these things? I have already written the answer somewhere here. Can you find it? Can you find the reason why a cell rapidly increases in size? What's the most possible, you have to select the best possible option. It's written somewhere here. So can you find it? I'll wait. Government Visa School, Pratima, Daya, can you answer this? And Vankir, can you try a bit? The answer is somewhere written over here. There is a hint over here. Can you find it? At least give it a bit. Try something. You can write maybe plastid. You can write the reason, maybe cell wall, cell vacuole, or plastid or something. But can you find the correct answer among this? What's the reason why a cell rapidly increases in size? What's that cell organelle which is responsible for that? Itanagar, Shillong, Tamenglong, Lunglai. Second point. Rapidly dividing cells. No, that's not the answer. That's not the answer. Swagata, can you answer that? Okay, I believe you do not have an idea about this. Okay. Look, cell vacuole I have written is absent or very much reduced. Cell vacuole is, you have seen a plant cell diagram, okay? Let me draw it. Okay. So you have seen a diagram of a plant cell from the initial stages. Okay, do not draw this diagram, just try to learn something from it. This is a conceptual point. The outer part is the cell wall, and what I'm drawing here, it is the plasma membrane or the plasma lemma. Now you have to identify all these cell organelles, okay? Nucleus, okay, it's fairly evident. Now this is, what is this? It is the mitochondria. And chloroplasts, stacks of coins, okay? So, alright, ribosomes, and this is the cell vacuole. Now you can see it's occupying the maximum portion of the cell volume. A vacuole, Government Visual will explain, a vacuole occupies a large part of the cell volume, even more than the nucleus. Okay, so you can say that cell vacuole is the maximum sized portion of maximum sized cell organelle of the plant. So vacuole, it is absent in the shoot apical meristem. Now the shoot apical meristem is going to convert into an elongated cell, a large cell. So which organelle must be present now? The cell vacuole is going to form, and because of the growth in the cell vacuole, the whole cell will increase in the size. Okay? This is the answer. So cell vacuole is responsible for the rapid increase in the size. Okay, that was what I was expecting from you, but no problem. Okay, so let me continue further. Okay, shoot apical meristem, you understood this point. Okay, so what is the use of shoot apical meristem? What is the use of shoot apical meristem? It has an application. Shoot apical meristem has a very interesting application. Whenever we have to perform tissue culture, we have to take virus-free portions from the cell, disease-free, bacteria-free, virus-free, pathogen-free portions. And at that point of time, we prefer to take parts from the shoot apical meristem. The meristematic zone, that part is what we prefer. The reason is because it's a rapidly growing cell, so if there is any diseased part, it must have already been degenerated or left behind. So shoot apical meristem always consists of healthy, rapidly dividing cells. So we always take a tissue culture sample for a plant from the shoot apical meristem. Why not from the root apical meristem? We can also take root apical meristem, right? We can also take root apical meristem for performing tissue culture. You know tissue culture, right? You take a small part of the plant, put it into growth medium, it will grow into a full plant. For example, I can take a carrot leaf cell, growth medium, agar growth medium. It will grow into a large mass of cells, and it will rapidly divide, and it will form a full carrot plant. So the thing is, which part will you prefer? You will take shoot apical meristem only. Why not root apical meristem? Root is in the soil, you can understand large number of microbes, pathogens, etc. will be present. So we will not take the root apical meristem. Okay. Shoot apical meristem is preferred material. Shoot apical meristem is preferred material for tissue culture. For tissue culture. Okay. Any plants that you know that are grown by tissue culture? Any plants around you You know that are grown by tissue culture. Do you know any such plants? Banana plants. Banana plants which are prepared by tissue culture, they have the ability, no, rose plant, not tissue culture, okay. Commercial banana plants which are available from the nurseries, from the market. Tissue culture, there are some plants, there are some strains which start giving the bananas in only 3 months. If you try to grow a banana tree, it will take about 6 months for it to give flowers and fruit. But if you take the tissue culture samples, it will start bearing fruit very early, within 3 months. Okay, so around you, you can see tissue culture applied in case of banana plants. Okay, all these are examples that will help you remembering all these things. These are tough things to remember, but you must remember at any cost. Okay, I'll give you some revision questions for all this, okay? So do not worry about that. So a limited time is present. I'll do what I can right now. Shoot apical meristem is done. Now let me cover the root apical meristem. Root meristem. Okay. So I told you root meristem. Where is it present? Let me draw root here quickly. Root apical meristem is present at a subterminal portion. It is covered by or protected by a root cap region. I'll draw this by a different color. Okay. Yeah. So this red colored part here, it is the root cap. It is the root cap. Okay. And just below it, what is present? It is the root apical meristem. Okay. Just above root apical meristem are the cells which are newly formed from root apical meristem. This means that these cells, they are newly formed. Okay, these are called. This is not a particular very short region, so we will call it a zone of root apical meristem. Sorry, root elongation. Okay. So which cell organelle will be present in zone of root elongation, and which cell organelle will be absent in root apical meristem? Cell vacuole, because these are rapidly increasing in size. And root hair will be present in which zone? The root maturation zone. This zone. Okay. Zone of maturation, also called the root hair zone. Okay. So root meristem, it is present in subterminal position. It is present at subterminal position just below the root cap. Subterminal position just below the root cap. Now tell me this, what will happen if the root cap is damaged? What will happen if the root cap is damaged? If the root cap is damaged, can you guess? We'll have to make a new root cap, okay? If you lose a helmet, you buy a new one. So similar is for the plant. If the root cap is damaged by the soil particles or by pathogens or by nematodes or by insects, the root apical meristem, it will form a new root cap. The cells of root cap are different from root apical meristem because root apical meristem is soft. Root apical meristem is soft, but the root cap zones cells are quite hard. So they will protect the apical meristem. Okay. So root cap is having protective function, and root cap cells, if they die, if they are damaged, they will be replaced by the root apical meristem. Okay. And zone of root elongation, I have only a limited time now. Root elongation, cells rapidly increase in length. Cells rapidly increase in length, okay, or volume. Okay. Next thing now, lateral meristem. Let me cover intercalary meristem because in this time only intercalary meristem might be covered fully. In the next class, I will cover the monocot, dicot, root stem sections and I'll explain that, and I will cover the secondary rows. So in next class, this chapter will be finished. Okay. So I'll give you the complete notes by then. Intercalary meristem. It is present between internodes. Okay. Suppose this is a stem. Okay. So this is a node. So this intercalary meristem, it will be present somewhere here. This is intercalary meristem. This diagram will also be in the notes. Okay. Or you can just see the NCERT diagram or figure. It is present between internodes. It is found in monocot grasses. Monocot grasses. Okay. Function is to rapidly increase in length, rapidly increase the length of plant in case of damage. Okay. Who will cause the damage? The damage might be caused by herbivores or by us when we use the lawn mower. Okay. In the case of damage. Okay. Why it has to replenish this damage so fast? Because it has it does not have a very good storage organ, so it has to make up for that damage very rapidly by performing photosynthesis, and photosynthesis performed by the stem. So to increase the length of the stem, it will increase the photosynthetic effective surface area. Okay. So this is about that. All right. So intercalary meristem. Now lateral meristem needs some discussion here. I need to explain a few things here. Lateral meristem is a complicated thing. I do not wish to complete it right now. Try to understand it like this. There are a few things you need to learn. First term is differentiation. Another term is de-differentiation. Third term is redifferentiation. Just try to learn these terms here because I don't believe I will be able to finish it. The first term I have talked about is differentiation. Differentiation means, in very simple terms, that a cell, a meristematic cell, has undergone cell division. A new cell is formed. Now this new cell will take its decision whether it has to become a part of parenchyma, collenchyma, sclerenchyma, or a part of xylem or of phloem. Now at this point, suppose this cell has become a parenchyma cell. Okay. But after some time, there is an event, a drastic event happens. Now this cell must divide. It has to divide. Okay. What are the conditions for this? What are the conditions for such a thing to happen? Suppose a plant has undergone injury. Okay. Suppose it was a parenchyma cell or a collenchyma cell and it has undergone injury. Okay. So the tissues have been cut off, a branch has fell off due to wind or by human action, something like that. So there is a wound here, an open wound is there, the cell sap is leaking, the plant sap is leaking out, and it is a good place for the fungus, for the bacteria, the pathogen to invade the plant. Okay, it's an open wound. We must heal this wound. So how the healing will happen? This cell, parenchyma cell, which is present there, it will suddenly undergo de-differentiation. Differentiation means a cell has chosen its carrier path. Okay, a cell has suddenly chosen its carrier path, it has become something, but now the need arises, it must undergo de-differentiation and must rapidly divide itself to fill this space, to fill the wound. So at such conditions, or in case of cambium cells, this cell which has undergone some differentiation, it has again become a meristematic cell. This is called de-differentiation. Differentiation means meristematic cell converted into permanent cell or parenchyma cell. Parenchyma cell converting into meristematic cell again. This is called de-differentiation. Now suppose the cell has divided. Now from the outside it must cover this area with a waterproof substance. This waterproof substance is called cork. Okay, so this waterproof substance is called cork, and some cells will form this substance and they will die. Okay, they will die because this is a waterproof substance. It has naturally only one fate. It must die. It cannot transport water. It cannot transport the materials required for the survival. So the outer cells are going to die, but they will protect the inner cells. Okay, so this type of cell which had become a meristematic cell now undergoes again differentiation into a cork cell or a bark cell. Okay, so this is called redifferentiation. Redifferentiation. A permanent cell has undergone de-differentiation, become a meristematic cell. Now meristematic cell has stopped cell division and it has again become a permanent cell. So three terms: differentiation, de-differentiation and redifferentiation. Okay, so try to understand it like this. Somebody is in class 10th and he has chosen a career path. He has chosen biology. Okay, and after studying about 1-2 months in biology classes, he feels very bored and he decides that I cannot bear this anymore. I must take mathematics. So he talks to the principal and he pleads to his parents and everyone who wanted him to become a doctor or something, and they push him back. He has undergone de-differentiation and now he has again taken another subject. He has taken let's say maths or maybe commerce. Okay, let's not take maths. He has taken commerce. Okay, so he was a math student. He was an undifferentiated cell. He had taken biology. Now he has taken commerce. So first thing which has happened is differentiation. The next thing it is de-differentiation. The third thing which has happened is redifferentiation. Maths to biology, biology to neutral, and then to commerce. So these things you will have to cover. I'll just write them down here. These terms must be discussed in lateral meristem. The first thing is differentiation. So what is differentiation? A meristematic cell, wait a second. A meristematic cell converted into permanent cell. Okay, this is a separate thing. You must understand this concept to understand about lateral meristem. So put it in a box or write it on your margin, top page. Meristematic cell converted into permanent cell. This is called differentiation. Differentiation. Okay. The next thing which has happened is it has undergone the decision to again become a meristematic cell. So this is called de-differentiation. Okay. De-differentiation. So it has become a meristematic cell. Okay. Now it has undergone integration. Okay, or it has become a permanent cell again. So this is not integration, this is redifferentiation. Okay, so these three terms you need to understand: differentiation, de-differentiation, and redifferentiation. I'll have to discuss this when I discuss secondary growth, because there are some cells which were permanent cell, they became meristematic cells, and they are responsible for the increase in thickness. So these three terms, whenever I take the next class, these must be very clear to you, because then I will use these terms rapidly, and please try to just understand about this, okay? So I believe this is, I will leave this here, okay? So thank you everyone, and see you in the next class, okay? And then we'll cover secondary growth and the cross sections, etc. Okay? Thank you, and have a great day.